Welcome, dear viewer, to our program, Knowledge Quest. The subject is biology. I'm your teacher, Lydia Asiago. In our previous lesson, we discussed the role of the kidney in excretion. In today's lesson, we are going to discuss the role of the kidney in osmoregulation. The role of the kidney in osmoregulation. Now, we ask ourselves, what is osmoregulation? When we define the word osmoregulation, is the balancing the amount of water and the amount of ions in the body fluids. Balancing the amount of water in the body fluids, balancing the amount of ions, e.g. salt ions like sodium, they usually affect the osmotic pressure. So when we talk about osmoregulation, we are balancing the osmotic pressure of the body fluids. Now, we have body fluids that surround the cells, body fluids, e.g. tissue fluid. We also have blood. Now, these are examples of body fluids in which we have to balance the osmotic pressure, because the osmotic pressure usually affects the functioning of the cell. Remember, if we don't have a conducive environment for the cells to function well, then it means we'll affect the whole functioning of the body. Take an example of an animal cell. So if this is an animal cell, this is just a sketch, and then in the animal cell, we have the cytoplasm, we have the nucleus, and we have the cell membrane. Now, take a situation whereby the osmotic pressure within the cell is higher than the surrounding. If the osmotic pressure within the cell is higher than the surrounding, it means the cells will draw water by osmosis. This being an animal cell, we expect this cell to swell and eventually burst. Why will the animal cell burst? Remember, it lacks the cell wall. So the cell membrane is not strong enough to withstand a higher osmotic pressure. So that's why we are saying that within the cell, we must have a conducive environment, and outside the cell should also have a conducive environment. Now, I give you an example. If there is a higher osmotic pressure within the cell, then it means the cell will draw water by osmosis, swell, and burst, meaning the functioning of the cell will stop. What if there is a higher osmotic pressure outside the cell? Then it means the cell will lose water by osmosis, it will shrink, and the normal functioning of the cell will also stop. And for that reason, we must maintain a constant osmotic pressure within and outside the cell for it to function well. And that is why we want to discuss, how does the kidney play a role in controlling or maintaining a constant osmotic pressure? Now, the kidney does this by the use of two hormones. We have two hormones that play a role in osmoregulation. Remember, I've said osmoregulation, we want to control or to have a constant osmotic pressure, an optimum environment for the cells to function well. Now, when we have a higher osmotic pressure within the blood stream, remember we said that we have the kidney this side, and then we say that the kidney receives blood from the renal artery, which subdivides to form the glomerulus, the afferent arteriole, which forms the glomerulus filtrate, the glomerulus, which also, where we have ultrafiltration taking place, you remember that? And we said this is the point whereby we form the glomerular filtrate, which flows all the way to the proximal convoluted tubule. And then we have the descending loop of Henle, and then we have the ascending loop of Henle. Now, you realize that at the descending loop of Henle, this is where we said that water is being reabsorbed by osmosis. But what influences the water to be reabsorbed? Number one, I said we have a hormone that is secreted by the pituitary glands. And that hormone, we said it is called antidiuretic hormone. Antidiuretic hormone is secreted by the pituitary glands. So the question is, how does it play a role? When the osmotic pressure within the cell, let me give an example, within the blood. Remember we said that we have blood vessels, and these blood vessels are carrying blood. When the osmotic pressure within the blood is higher, above the normal, the part of the brain known as hypothalamus. The hypothalamus is stimulated. Hypothalamus is a part of the brain that is responsible in osmoregulation. It's also responsible in thermoregulation and the regulation of blood sugar. That is the role of the hypothalamus. Mostly it plays a role in homeostatic function. And that is why I want to explain how does the kidney play a role in osmoregulation. Number one, when the osmotic pressure in the blood, assume these are blood vessels, and inside the blood vessels we have a higher osmotic pressure. The hypothalamus will be stimulated. And the moment the hypothalamus is stimulated, it will send an impulse to the pituitary gland. The pituitary gland will release antidiuretic hormone. This antidiuretic hormone is released into the bloodstream. So the moment the antidiuretic hormone reaches the kidney tubules, it makes those tubules to be permeable to water. So as a result, more water is being reabsorbed back into the blood. And when you reabsorb more water from the glomerular filtrate into the blood, then you are going to lower the osmotic pressure. Remember, we are discussing the role of the kidney in osmoregulation, regulating the osmotic pressure within the cells, within the body fluids, because we need the cells to have an optimum environment for the normal functioning of those cells. And I've said, when there is a higher osmotic pressure within the blood stream, the hypothalamus is stimulated. It sends an impulse to the pituitary gland, which releases a hormone known as antidiuretic hormone. The antidiuretic hormone is released into the bloodstream. On reaching the kidney tubules, it increases the permeability of the kidney tubules to water, meaning more water is reabsorbed back into the blood, hence lowering the osmotic pressure. Now, when the osmotic pressure is lower as compared to the body, the osmotic pressure in the blood is lower as compared to the surrounding. What happens is that the pituitary gland is less stimulated. So we expect the hypothalamus to send an impulse to the pituitary gland to release less antidiuretic hormone, meaning when you have less antidiuretic hormone in the bloodstream, then it means less water is being reabsorbed. So there are two situations. There are times that you find someone produces out large quantities of dilute urine, and at times someone produces little quantities of urine that is highly concentrated. So under what conditions do we produce that? Number one, when you have a higher osmotic pressure, higher osmotic pressure in the blood. Of course, we said hypothalamus is stimulated, sends an impulse to the pituitary gland. The pituitary gland releases the antidiuretic hormone, which is released into the bloodstream. On reaching the kidney tubules, it increases the permeability of the kidney tubules to water. Hence, more water is reabsorbed back into the blood. So when there is a higher osmotic pressure in the blood, then we expect someone to produce less urine, and this urine is highly concentrated. Highly concentrated. So those are the conditions whereby you find someone producing less urine that is highly concentrated. Why? Because in the body fluid, especially in the blood, you have a high osmotic pressure, and as a result of that, your pituitary glands are releasing a lot of antidiuretic hormone, which is increasing the permeability of the kidney tubules to water. Hence, more water is reabsorbed back into the blood. And that is why you are releasing less urine, and this urine is highly concentrated. Now, what happens when the osmotic pressure in the blood is low? When you have low osmotic pressure in the blood. Remember, I'm comparing the osmotic pressure in the blood and especially in the glomerular filtrate. Remember the glomerular filtrate, that is where we have a lot of water that came out of the blood as a result of ultrafiltration. So when there is a low osmotic pressure in the blood, the hypothalamus is stimulated. It sends an impulse to the pituitary gland, and the pituitary gland releases less antidiuretic hormone, and as a result, less water is reabsorbed back into the blood. So the urine that someone is passing out large quantities of urine, and this urine, of course, is dilute. And then we say we dilute. The urine is dilute. Why is it dilute? Of course, it has a lot of water, and that water is the one that dilutes. Of course, when you have a solution consists of a solvent and a solute. So when you have more solute, it means that solution is concentrated. When you have more solvent, it means that solution is dilute. And that's why I'm saying that when you are passing out large quantities of urine, it means there is a lot of water in that urine, and that's why we are saying it is dilute. Now, there is a situation whereby someone fails. The pituitary gland fails to produce enough antidiuretic hormone. What happens? It means that less water is reabsorbed back into the blood. As a result, this person passes out large quantities of dilute urine. Such a person is not normal. Such a person is suffering from a condition we call diabetes insipidus. And that condition is whereby someone feels thirsty all the time. Diabetes insipidus. It is a condition whereby the pituitary gland of someone is not producing enough antidiuretic hormone. So as a result, you find that the kidney tubules are not able to reabsorb back most of the water. So you find that much of the water comes out in form of urine. And because of this, this person feels thirsty all the time and drinks a lot of water. So the only way to compensate for that condition is you take in a lot of water. Or you can go and see the doctor if they can initiate your pituitary glands to produce more antidiuretic hormone. Now, of course, we can call it a disease. We can also talk about the condition. The condition, we call it diuresis. Diuresis means someone feels thirsty all the time. You drink a lot of water, but still you feel thirsty. Why? Much of this water, after ultrafiltration takes place, much of this water does not come out of the filtrate back to the blood, but it goes direct out in form of urine. Now, this is how the kidney is able to control the osmotic pressure within the blood. Now, remember I said osmoregulation means you are balancing the amount of water in the body fluids and the amount of salts. So osmoregulation involves two aspects whereby we are balancing the amount of water in the body fluids. At the same time, we are balancing the amount of salts. There are salts that are found in the body that are very, very important. Example, we have sodium ions, we have potassium ions, we have calcium ions, we have phosphate ions. All those salts that are found in the body fluids, especially in the blood, they also affect the osmotic pressure. The kidney also plays a role in balancing the amount of these salts in the blood. For example, we ask ourselves, do we need sodium ions? Yes. Do we need potassium ions? Yes. Where do we apply them? For example, potassium and sodium ions, they are used in nerve transmission. Calcium ion is also very important in blood clotting. So we need to balance the content, the amount of these ions in the blood or in the body fluids because they affect the osmotic pressure. Now, the kidney does this by the use of a special hormone known as aldosterone. Aldosterone. Aldosterone is a hormone that is produced by the adrenal glands. How does the kidney regulate the amount of these ions in the blood or in the body fluids? Number one, we know that when you have high concentration of these ions, then it means you increase the osmotic pressure. When you have low concentration of these ions, the osmotic pressure is low. I want to give you a scenario. Take a situation. When you have a solute plus a solvent, you form a solution. I'm giving you this scenario so that you try to picture that we must balance the amount of solutes and the amount of solvents that we have in the body. Now, in this case, let me pick an example of the solute as sodium. So if you have salts plus water as our solvent, then we are going to form salt solution. Salt solution. Remember, when these salts are absorbed into the bloodstream, usually they dissociate, especially if you absorb sodium chloride. The moment it gets into the bloodstream, it dissociates into the ions, sodium ions and potassium ions. Now, take a situation that in the blood you have more salts and you have less water. So in this case, water is our solvent. Then which solution are we going to form? When you have more salts, then we say the solution is highly concentrated. So we say this is highly concentrated. Now, when you have less salts and you have more solvent, which is water in this case, then we say the solution is lowly concentrated. Now, there are two concepts I want you to understand. When we say a solution is highly concentrated, it means there is a higher osmotic pressure within that solution. When we say the solution is lowly concentrated, then it means within the solution there is a low osmotic pressure. Now, if you can picture that, so we say highly concentrated solution, then it means there is a higher osmotic pressure. Higher osmotic pressure. And if you have a solution that is lowly concentrated, then it means lowly concentrated solution. Then we say this solution is having a low osmotic pressure. Low osmotic pressure. Now, if within the blood you have a highly concentrated solution, like the osmotic pressure in the blood is higher, then it means you have more salts. And because you have more salts in the blood, then it means the hypothalamus will be stimulated to send an impulse to the adrenal glands to release less aldosterone. So when you have a higher osmotic pressure in the blood or in the body fluid, then you have less aldosterone. Less aldosterone means there is less reabsorption of mineral salts in the descending loop of Henle. We are going for a short break. Stay tuned. We'll be back. Welcome back, my dear student. Before we went for a short break, I was trying to explain you to understand the concept of a higher osmotic pressure and a lower osmotic pressure. Now we say when a solution is highly concentrated, then it means we have a higher osmotic pressure. When the solution is lowly concentrated, then it means you have a low osmotic pressure. And for the kidney to function in regulating the osmotic pressure, we said that there are two hormones that are playing a role. The first one is antidiuretic hormone, and the second one is aldosterone. Antidiuretic hormone regulates the amount of water in the body fluids. Aldosterone regulates the amount of salts, amount of ions in the body fluids. Now, when we have a low osmotic pressure within the blood, low osmotic pressure, meaning the solution, the content in the blood is lowly concentrated, then it means the hypothalamus will be stimulated to send an impulse to the adrenal glands. Remember we said that adrenal glands, they are the ones that produce aldosterone. So the hypothalamus will be stimulated to send an impulse to the adrenal glands to release more aldosterone. Aldosterone will be released into the bloodstream. On reaching the kidney tubules, remember we said that at the descending loop of Henle, that is where we have active reabsorption of mineral salts, e.g. the sodium ions. So we expect the kidney tubules to be more permeable to mineral salts like sodium. So when we have a low osmotic pressure in the blood, then it means the hypothalamus is stimulated, sends an impulse to the adrenal glands. The adrenal glands releases aldosterone. The aldosterone increases the permeability of the kidney tubules to ions, e.g. sodium ions, meaning more salts, more sodium ions will be reabsorbed back to the blood. Remember we said the moment you have more salts in the blood, then again you are going to have a highly concentrated solution, meaning you are going to raise the osmotic pressure. So the moment the osmotic pressure increases, of course, above the optimum, what happens? The hypothalamus is less stimulated and it sends an impulse. When the hypothalamus is stimulated, it sends an impulse to the adrenal glands. What is the message? Produce less aldosterone. And when you have less aldosterone, it means less mineral salts are reabsorbed back to the blood. As a result of that, again, you are going to have a situation whereby you have what? you have less salts in the blood and you have more water. Meaning, again, in the blood, it becomes lowly concentrated. So you see there is a balance. We want to balance when the osmotic pressure goes below the optimum, it has to be raised. When the osmotic pressure goes above the optimum, it has to be lowered. And if you are very keen, you realize that aldosterone and antidiuretic hormone, they work hand in hand. When you have more aldosterone in the bloodstream, you expect to have less ADH. Why? When you have more aldosterone, then it means you need to reabsorb more salts, because at this point, when you have more aldosterone, you have less salts. So you need to reabsorb more salt and reabsorb less water. Why? Already you have more water in the blood. When you have more antidiuretic hormone released in the bloodstream, then it means you have less aldosterone. Why? More antidiuretic means you have less water in the blood and you have more salts. So you need to reabsorb back more water but less salts. And that is how the kidney plays a role in osmoregulation. Now, the next question. Is the kidney the only structure that is used in excretion? Is it the only organ that is responsible in excretion? No. Remember, we listed, from the beginning, we listed and said that we have 4 organs that are responsible in excretion. Of course, excretion and homeostasis. Number 1, we mentioned the kidney. We also mentioned the liver. We mentioned the skin and the lungs. Those are 4 major excretory organs that we have in a mammal. Now, having done with the role of the kidney in excretion, the role of the kidney in osmoregulation, I want to move on to the next excretory organ that is very, very important. That is the liver. So we want to discuss on the liver as another excretory organ. The liver is the second largest excretory organ that we have. First one being the skin. The skin is the largest, followed by the liver. Now, how many roles does the liver play? Number 1, the liver does the role of excretion. That is the first role. Number 2, the liver does the role of thermoregulation. Thermoregulation. Number 3, the liver does the role of balancing the amount of sugars in the blood. Regulation of blood sugar. For now, I will discuss the 3 major functions of the liver. That is excretion, thermoregulation, and regulation of blood sugar. I'll start with the first one, excretion. How does the liver play a role in excretion? Number 1, the liver does excretion in many other ways. It does through deamination. Remember, we said that excretion, we are releasing substances from the body that are formed from metabolic processes, and those waste products, we have to release them. So the liver does the role of excretion, number 1, by deamination. Number 2, by detoxification. I think I'll discuss the 2 first so that we see how does the liver play a role in excretion through the process of detoxification, through the process of deamination. I'll start with deamination. What do we mean by deamination? You remember, the liver receives blood through two vessels. Number one is through the hepatic artery. Hepatic artery is the one that carries blood from the heart to the liver. And the blood in the hepatic artery is full of oxygen and any other excretory product that needs to be excreted. The other second vessel that supplies the liver with blood is the hepatic portal vein. Hepatic portal vein carries blood from the stomach, that is from the small intestine, into the liver. And the hepatic portal vein, number one, it is having blood rich of all useful substances that are formed after digestion. Remember, during the process of digestion, all carbohydrates are broken down to simple sugars, e.g. glucose. All the proteins are broken down into amino acids. All the lipids are also broken down into fatty acids and glycerol. So once all these substances have been absorbed into the bloodstream, they flow to the liver. And there's a reason why they flow into the liver, especially for the case of simple sugars, which are osmotically active, like glucose. It has to be controlled. You must have an optimum amount of glucose in the blood. Reason being, glucose is osmotically active and it affects the osmotic pressure in the blood. And that's why we are going to discuss why we should have regulation of blood sugar. It is because sugar is osmotically active and it will affect the osmotic pressure within the cells. And remember, we say that osmotic pressure within and outside the cells should be at a given optimum level for the proper functioning of the cell. Now, when the blood from the stomach is carried all the way to the liver through the hepatic portal vein, this blood, number one, is full of amino acids. Amino acids. Now, amino acids, these are product of breaking down proteins. And the moment we absorb the amino acids into the body, we use the amino acids to synthesize proteins. But the question is, where does the excess go? Our bodies do not have a mechanism of storing excess amino acids. So the excess amino acids are eliminated from the body system through the process of deamination. And this process takes place in the liver. So briefly, I want to explain how does deamination take place as a process of excreting excess amino acids. If you look at the structure of the amino acid, I believe you know the structure of the amino acid. The amino acid, we have the carbon, we have the amino group. This is the amino group. Then we have hydrogen. We also have the carboxyl group. And we have that one. Now, during the process of deamination, I'm explaining the process of deamination. During the process of deamination, the amino group is removed and it is converted into ammonia. So, amino group that is in the structure of the amino acid is removed and converted into ammonia. Ammonia gets into a cycle that consists of a series of reactions. We call it on-time cycle. Let me write that so that you are able to follow the process of deamination. We said that during deamination, this is the structure of the amino group. The structure of the amino acid. And the amino acid has the amino group. Now, we are saying that during the process of the deamination, the amino group is removed and converted into ammonia gas. Now, the ammonia gets into a series of reaction we call on-time cycle. Now, so if I have ammonia, this is ammonia, it combines with carbon 4 oxide in a series of, we say, on-time cycle, whereby it is a series that is controlled by so many enzymes, and there is a lot of reactions that takes place in this process. And when carbon 4 oxide combines with ammonia, it forms urea plus water. And of course, we know that from the previous lesson, we discussed those metabolic wastes, and we said that when you compare ammonia and urea, ammonia is more toxic. It is highly soluble in water, hence requires a lot of water for its elimination. But when you have urea, we say it is less toxic, it is less soluble in water, and it requires less water for its elimination. And that's why we are saying that during deamination, we are able to excrete excess amino acids by removing the amino acid, which is converted into the ammonia, and the ammonia combines with carbon 4 oxide in a series of reactions known as the on-time cycle to form urea and water. And now this urea is the one that is going all the way to the kidney for it to be excreted. Remember, we discussed the role of the kidney in excretion, excreting urea in form of urine. So our urea is formed in the liver. So we expect from the liver, through the hepatic vein, the urea should flow all the way to the heart. Then from the heart should be pumped to the kidney where it is excreted. So that is one process through which the kidney plays a role of excretion, through deamination. And I've said deamination is a process whereby the body excretes only excess amino acids. Why? It is because the body does not have a mechanism of storing excess amino acids. Now, another way through which the liver plays the role of excretion is through detoxification. Detoxification means you are converting a toxic substance into non-toxic. And that is one of the roles of the liver, detoxification. We are going to give an example of a process whereby we are able to see detoxification taking place. Detoxification. Now, during detoxification, you realize that the more toxic substances are converted into non-toxic. So toxic substances are converted to non-toxic substances. Now, how does this take place? Take a situation at the respiring cells. You remember we discussed the process of respiration. And we say that during respiration, we produce energy, water, and carbon 4 oxide. But at times when the cells are actively respiring, instead of them producing water, they produce what we call hydrogen peroxide. Let me give an example. Now, when you talk about hydrogen peroxide, is an example of a toxic substance that is produced by actively respiring cells. At normal situation, we expect when glucose is broken down, glucose, oxygen plus oxygen, we are supposed to have water plus carbon 4 oxide plus energy in form of ATP. But now, those cells that are actively respiring, instead of them producing water, of course water we say is HO2. Instead of producing water, those actively cells, especially the liver cells, they produce hydrogen peroxide. But the liver cells consist of an enzyme catalase which breaks down the hydrogen peroxide into water and oxygen. Now, you see the products that are formed, they are not toxic. Hydrogen peroxide is toxic, but water is not toxic, oxygen is not toxic. And because of that, we are saying that the liver does the role of detoxification, converting the toxic substances into non-toxic or into less toxic. And I've given an example of the actively dividing cells, actively respiring cells in the liver. Instead of producing water, they produce hydrogen peroxide. But the liver is able to break down hydrogen peroxide into water and oxygen because it has an enzyme known as enzyme catalase. So I've given you two examples on how the liver plays a role in excretion, removing excess amino acids through the process of deamination and eliminating the toxic substances through the process we call detoxification. Now, we are going to see how does the liver play a role in regulating the blood sugar. Remember we said that the blood sugars, they are osmotically active. They affect the osmotic pressure of the blood. And if they affect the osmotic pressure of the blood, it means they will affect the normal functioning of the blood, especially when you have a higher osmotic pressure or when you have a low osmotic pressure. So the next function of the liver that we are going to see is the regulation of blood sugar. An example of blood sugar that we have is glucose. Now, in our next lesson, we are going to discuss the role of the liver in regulating the blood sugar, which mostly affect our way of life, and how should we have it at an optimum level whereby it doesn't affect the osmotic pressure of the cells. We need a conducive environment for cells to function. We don't want cells to have a high osmotic pressure as compared to the environment because they will draw water, swell, and burst, meaning the functioning will stop. We don't want the environment of the cells to be highly concentrated as compared to the contents in the cell. Why? The cells will lose water, shrink, and stop functioning. So because of that, we need to regulate the amount of blood sugar that we have in our bodies. But who does that? It is the liver. In our next lesson, we'll discuss the role of the liver in regulating blood sugar in our body. Thank you for watching. I've been your teacher, Lydia Asiago.